Hello, good afternoon. Good afternoon, everybody. And probably to end the session, as I wanted to just tell you a small story about citizen scientist involvement in documenting biodiversity in India. Uh, Throughout the world and including in this JBS7, there were several presentations, including posters on involving citizen scientists in documenting uh, jellyfish diversity across the world, monitoring ecological interactions, and as well as even in genetic studies. And let me start with the 10 commandments in citizen science programs throughout the world, and whether this presentation fits into that. And one is that we involve them in all these scientific endeavors. And second, it should have a genuine science outcome. And third, the professional scientists should involve the citizen scientists, and there should be mutual benefits. And the fourth, they participate in multiple stages in the whole process of research methodology, right from framing the hypothesis to finalizing the results and conclusions, and whether they receive feedback from the scientific community, and that's the fifth one. And the sixth one is the citizen science is considered as a research approach like any other. The whole process involved the research process. And seventh one is, the data and metadata is made available publicly in the form of a research paper later. And the eighth one is citizen scientists are acknowledged in project results and publications, and that's another component. And citizen science programs are evaluated for their scientific output, data quality, participant experience, and wider social and policy impact. And the final commandment, which is again, all these commandments were proposed by the European, the Citizen Science Association. And there are whether some legal or ethical issues are involved in this. And we have seen in the last few days there are large number of successful projects across the world involving many citizen scientists groups, starting with the Jelly Watch to many other. I am not naming all. And all these things are involved. And when you look at these successful citizen science programs in documenting jellyfish diversity, we would understand that that is highly skewed. And there are only two stronger representation from Asia, and one is in primarily by the Israeli scientists, and the second from the Hong Kong group. And there is no much representation elsewhere. I just wanted to tell you why it is important, and especially in Indian context. And I start the story when we involved a citizen scientist, and his name is Umid. Umid, are you here? Yeah, Umid is here. And I wanted to also to acknowledge him because most of the photographs you see in the photography exhibition is taken by Umid. And actually, I don't know him earlier, and then we were actually involved him in installing a hydrophone to locate the movement of whales along the Kerala coast, and he was the person who was helping us. And then when we started the launching this program JBS7, I asked him to give some good photographs, which we can use it in the website as well as in the brochures. And then he gave the photographs, and then we started identifying them using he dive across the world, in fact, and then some of the dives in India provided some good quality photographs, including the amphibious photographs which you see there, but that's not from India. And the first one is the dive from Andamans, and Andamans from the Andaman Islands, as you can see there on the map there. He photographed this Aurelia species, and when we started analyzing along with the Rias, and we found that the structurally this species is slightly different. And because in India we had Aurelia aurita and Aurelia solida reported earlier, but this animal has some strikingly different characters, particularly in its lappets, and they have more than 8 lappets, more than usual lappets. and that led us to some molecular work which is now going on. And probably the species which bloom, as you can see in the beautiful photograph by Umi, is that this could be a new species which is seasonally bloom in Andaman waters. And this is another species. Again, the photographs, the quality of the photographs is so high. Messages Papua, and this is from the Pak Bay in Tamil Nadu. And also some of the a series of photographs. I'm not showing all the photographs. This series of photographs also tells us several stories regarding the ecology of this species. Particularly, it's the movement patterns and in the seagrass meadows in the region. And again, some of the very interesting things which probably we have to work on later. So all these photographs actually provide us some of the topics to work on in future. And this is Lobonima smithii, and this is again the species which occur in the coral reefs ecosystem in the Pak Bay between India and Sri Lanka. And this is one of the largest species, and you look at because we are impressed by the quality of the photograph. And the species is Ropilima hispidum. And then if you look at in detail, we could study the ecology as well. How the carangid, especially the Elypus jabada, and how they are associated, their number, and the way in which they associate with this organism, etc. So that is another interesting feature. And when Catacylus was photographed by Umid from Andamans in India. And this is again it could be the Catacylus mosaicus, and this is a new record to Indian waters, again from the citizen science photograph. And next beautiful photograph is Crambion mastigophora, and this is again from Andamans. Again, no record earlier from Andamans. Andaman is again a coral islands represented by more than 360 islands in Indian region. And this is Saina Nosaki, again by another fellow diver, Jonah, shot from Tamil Nadu coast, from Trivandrum coast, very close to that. And yesterday I've seen some of the social media postings by Stamper, where he also recorded the same species from some of the dive sites in Trivandrum. And some of the photographs which is exhibited here will also provide us some of the very interesting associations here. And this is Sainia Nosaki with the closely associated brittle star. Ophionemis marmorata. And this is a photo by Daniela, a fellow of Umid. And there's a specific reason why I showed this photograph here. And many of the species which are documented as Sainia nosaki from India are not Sainia nosaki. And this is the Sainia nosaki which she photographed from Maldives. Is it Maldives, Daniel? Yeah. It's from Maldives. That means, this is the species which is not present in India, but it is present in the Maldives. And so the involvement of the scuba divers in documenting the photographs as well as cataloging biodiversity along the Indian coast is very important. And I come with some of the suggestions, and primarily we need to involve the practicing, to help the practicing taxonomists, who are very few in a larger country like India, especially if you look at the jellyfish documentation in India. In still 1950s, there were stronger records of documentation, and only in the last one decade, the people again started working on it. And in many of the hydrozoan groups, there are no experts in India as well. So this is a time where we need to involve the citizen scientists in documenting diversity primarily, particularly in a context when the scientific funding agencies, and also I'll tell you that in India funding agencies are primarily the government agencies. Normally, throughout the world, this happens. The funding agencies, if you apply for a space science project, you will get it. In nanoscience, nanotechnology, you will get it. But for the basic sciences, it's hard to get money for running the projects or surveys. And though this is the only way in the post-COVID world we can involve citizen scientists, including as parataxonomists, to collect data and help the taxonomy community here to document the data strongly and build a stronger database over a period of time. And also, there are few departments, including the marine science departments, which work parallelly, and there are a lot of duplication of work. And I recommend again that this kind of duplication of work can be avoided by strategically planning marine exploratory surveys. And there is a stronger need to develop a group of youth primarily trained and in or skilled in taxonomic exercises, and that could be the aim of the workshop which is coming after this conference. And also we need a stronger the local field and survey manuals in this group. And also we cannot work in all the areas together. We need to prioritize the hotspots, especially in the knowledge gap areas. And finally, there is an example of involving the citizen scientists in documenting biodiversity in India. This could be one of the best models as far as India is concerned, and that is eBird. And when eBird was launched in India in 2014, and a lot of passionate birdwatchers joined it, free of cost. And it is now, after 5 years, when they initially published the data, and currently there are more than 1,300 species covered, and more than 1 million of checklists available from each and every part of India, a larger country like India, and more than 200,000 photographs and the calls recorded by various citizen scientists across the world, and more than 10 million observations in all. So this is a typical or classical example how we can involve citizen scientists in documenting biodiversity, but that is not happening in the marine environment as far as India is concerned. And my appeal is that it can happen in the marine side as well. And some of the international communities and groups which are already documenting it can collaborate with the Indian partners so that the documentation process can involve large number of scientists as well. And the reason is very simple, because we have a lot of technically trained, especially in IT and other sectors, and most of the youngsters in India are trained in IT because they get job there, and then they are also passionate in biology. So once we involve them and assign them with the different duties, including in cutting edge research involving jellyfish, and we can have a stronger database to present in JBase 8. Thank you.